Tubular reabsorption is the process of reclaiming water and solutes from the tubular fluid and returning them to the blood. There are two routes of reabsorption. The first is the transcellular route, in which substances pass through the cytoplasm of the epithelial cells by way of transport proteins in the cell's plasma membrane. The second is the pericellular route, in which substances pass through the gaps between the cells. The water carries dissolved solutes with it in a process called solvent drag. Water and solutes leave the tubular epithelium via the basilar surface and then enter the tissue fluid of the kidney. Then, water and solutes are reabsorbed into the bloodstream by the peritubular capillaries. This happens by way of osmosis and solvent drag. Three properties promote the movement of water from the tissue fluid into the peritubular capillary. The first is the tissue fluid hydrostatic pressure, which is high because of all of the water leaving the tubular epithelial cells. Second is the narrow efferent arteriole of the glomerulus. This lowers the blood hydrostatic pressure in the peritubular capillary. And finally, since proteins do not leave the blood in the glomerulus, but water does, the blood colloid osmotic pressure is elevated in the peritubular capillary. When you compare the osmotic and hydrostatic pressures between the tissue fluid and the peritubular capillaries, reabsorption into the capillary is strongly favored here, and therefore most of the water filtered out of the glomerulus is returned to the bloodstream. Now there's a limit to the amount of solute that the renal tubule can reabsorb because there are limited numbers of transport proteins in the plasma membranes. If all the transport proteins are occupied, some solute that normally is reabsorbed, like glucose for example, will escape reabsorption and appear in the urine. The maximum rate of reabsorption is called the transport maximum, and it is reached when the transporters are saturated.